Hey guys, today we're going to be talking about the catalase test. And the catalase test is used to differentiate bacteria based on the presence of the enzyme catalase. So this test is really good at differentiating between staphylococci and streptococci. The basics here is that if a bacterium utilizes aerobic respiration, it will utilize oxygen as the final electron acceptor, producing water. Now, along the way here, though, generally hydrogen peroxide is produced as a byproduct. And hydrogen peroxide is fairly toxic to things like DNA and RNA. So the bacteria will want to neutralize it, and that's what it uses catalase for. It can basically convert hydrogen peroxide back into water and oxygen, normally forming bubbles. You might have seen that if you put hydrogen peroxide on a cut or something like that, because we have catalase as well in our body. The way that this is used to differentiate between a staphylococci and a streptococci is that your staphylococcal species are obligate aerobes generally, whereas the streptococci are facultative anaerobes. So your staphylococci are going to go through and they're going to be producing catalase to sort of neutralize this reaction, whereas your streptococci, well, they don't utilize oxygen, so they don't really have a need for catalase. So it's an easy way to tell the difference between those two. Now, in terms of actually setting up the experiment is really straightforward, but let's just kind of run through the materials that you need to actually go through and do the test. So you are going to need a Bunsen burner for a transfer. You will need your striker to turn it on, and then the loop to actually transfer the bacteria. We are going to be transferring to slides, so you're going to need at least one slide. You're going to need a bit of hydrogen peroxide to actually do the test. So but hydrogen peroxide, I have a bit in my beaker here to make it easier to transfer it with the dropper and put it in the slide. And then you're going to need your sample. So this test generally works out better if you do it from a plate than if you do it from a broth. The main thing here is that if you're going to see a positive result, which is indicated by bubbles, the more bacteria you have, the more bubbles you're going to see. And it's easier to get a lot of bacteria from a plate than it is from a broth. So normally, again, we transfer from a plate. If we want to transfer from a broth, which we can do, you'll want to do a lot of transfers onto the slide before you add the hydrogen peroxide. But with that out of the way, let's kind of go through and run the test and see what we get. So let's go ahead and light our Bunsen burner here. Get our slide ready. I'll put it there for now. And let's go ahead and make sure that, of course, we transfer everything aseptically. Get our loop nice and sterilized. All right. So a good trick, again, to cool your loop down quickly is that when you go to grab your sample, find an area of the plate where there is no growth and just kind of tap it along that area. And then I'm just going to go in. I have a nice big colony here, so I'm just going to grab a nice big chunk of bacteria again. Generally, again, we're going to take a lot. I'm just going to put that on here. Kind of get that nice big clump on my slide. Go ahead and flame again to clean off the loop. Boom, 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 boom. Really wish there was a way to make this faster. All right, so we should be good there. Go ahead and turn this guy off. And then now, we'll go ahead and add a drop or two of hydrogen peroxide. All right, let's see, I did 3 drops there. And to kind of show you guys what we're looking at, try not to tilt it too much, but you can really see, again, there is quite a few bubbles that we have in there. So if you see bubbles, that is a good indication of a positive result for catalase. If you add the catalase and you see no bubbles, that's going to be a negative result. Now, the general instructions you're going to find in manuals and things like that is that it's going to say that you want to wait about 5 to 10 minutes before you record what you see. Generally, this reaction will be very quick. It will be almost instantaneous once you add the hydrogen peroxide. So especially for taking your pictures and things like that, make sure that you have your phone ready so that you can take the pictures and the bubbles don't, they're going to disappear after a while. There's oxygen gas that we're seeing, and it's going to dissipate into the environment. So make sure that you have your phone ready to take a picture basically right away of what it is that you're getting. And if you get a negative result, then you want to wait the 5 to 10 minutes. But that should really include everything that we need to know about the catalase test.